Hi, folks. I hope you're having a pleasant start to the week here. My guest today is Ryan Cochrane. He's a PhD student studying synthetic biology and specifically looking at algae and how to do whole mitochondria genome transformation into algae. And if that doesn't make any sense to you, well, that's okay because we're going to really get into it into this episode. For instance, I didn't know mitochondria has its own DNA, and I didn't know that the mitochondria splits separately from the cell itself. And uh, and so probably some of you have heard of mitochondria back in your middle school or high school biology days and know mitochondria as the powerhouse of the cell. But imagine if you could get control over the functions of mitochondria. Since mitochondria plays such an important role in the metabolism and the function of the cell, if you can gain a mastery over mitochondrial DNA, you can get the cell to produce really valuable things in a way that makes it more commercially viable and is a little bit less complicated than when trying to do the same thing through nuclear DNA. So we really got into it. Ryan is incredibly knowledgeable, obviously studying genetic engineering and synthetic biology at such a deep level. It's really a privilege of mine to be able to pick his brain about my journey in learning how to genetic engineer fungi. And so I learned a lot from this episode. I learned a lot about algae as well. Algae is really gaining a lot of popularity in the biotech community and amongst DIYers as well because it's a pretty easy organism to grow, and it's really just another interesting organism to look at and to consider when you're thinking about where to focus your energy. So it's pretty cool to learn about this other organism and then make all of these connections between algae and then the fungi that I'm learning how to engineer. So we really geeked out on this episode. I enjoyed it. And I think that you're going to really learn a lot about this stuff too. So thanks again to Ryan. And without further ado, Ryan Cochrane.
Ryan, thanks so much for joining me on the podcast. It's a pleasure to finally get a chance to speak with you after some back and forth on social media and email. So my understanding is you're studying synthetic biology right now. Is that right?
Yeah, I'm at the University of Western Ontario here in London, Canada, London, Ontario. And I work under the supervision of Dr. Bogomil Karish and David Edgel. Both of them have varying degrees of synthetic biology background, but both have recently transitioned their lab focuses in that direction, I feel like. Specifically, I work in the Karish lab mainly.
Karish lab.
And we focus on whole genome engineering or innovative strategies for developing innovative strategies for whole genome engineering. And I work with two different microalgae, and specifically their mitochondrial genomes. And so the focus of my thesis mainly up to this point has been cloning those genomes and developing a platform for engineering them.
Okay, cool. I mean, I definitely want to get into a lot of that because you threw around a lot of words that I'm not super familiar with quite yet. So,
yeah, very.
How many? So you're going for your PhD.
Yes.
What year would you, or how far into that process are you currently?
So I'm in my fourth year now.
Is that the final year, or could it take a little bit longer?
So with a direct entry PhD, it's typically 4 years. With that kind of route, you're looking at somebody who already has some research in the lab and familiarity with their project before they begin the program. And then they jump right in and hit their 3 chapters out kind of deal. I took a non-direct entry, so I started as a master's student. And after your first 1 or 2 years, you take this qualifying exam to transfer into the PhD program. But that comes with an additional year of schooling. And so I would typically end on a 5th year. So I have 1 more year after this.
Oh, wow. Okay. So you're making pretty good progress. You're just about done.
Yeah, we're closing in on the end for sure.
Yeah, that's really cool. And I think that I wanted to ask you about one of your publications because I think I came across maybe a paper or two that you'd recently published.
Yeah, I have 2 different manuscripts, one for each mitochondrial genome of 2 different algae, one being Phylodactylum tricornum and one Thalassera pseudonina.
Okay.
So it's just 2 different species names. That's all. Both are diatoms. Yeah.
We definitely will touch on the technical stuff because I'm really fascinated by that. But I kind of wanted to just kind of go back into your journey of how you arrived to where you're at today. Did you always know you wanted to do synthetic biology, or how did you find out about it?
Yeah, so I didn't go into university thinking I'd be going into synthetic biology or anything by any means. In high school, I kind of just wanted to pick the most difficult program I could find, which I had interest in. And so I had originally applied for like neuroscience, and that eventually transitioned into like a physiology and pharmacology degree, and then an interdisciplinary medical sciences. So it's kind of, the program touches on many different aspects of medical science. And one that really caught my interest was biochemistry. And so I went to the graduate student night where they recruit students locally. And I was the only one who showed up that year. And so I met my co-supervisor, Dave, who seemed to like me a little bit and told me to apply for grad school. And so I did. And I ended up with him. And he introduced me to Bogomil Karish, who was working in his lab at the time. He wasn't even hired as a PI. And so Bogomil was super enthusiastic about synthetic biology. He actually had just came back from well, not just came back, a year or two, but his most recent position was at JCVI, the Craig Venter Institute in California. And so he had already had a big grasp on synthetic biology. He was very much into it.
Yeah, that's pretty cool. And for people who don't know, because I hear a lot of words in synthetic biology used interchangeably. I hear genetic engineering, I hear synthetic biology, and then biochemistry, which is sort of more larger encompassing that
Yeah.
those things roll into. What are all of those things for people who've never heard of genetic engineering, or maybe they heard of it, they're not really sure what it is? How would you explain it to them?
Yeah, so genetic engineering, I would say, is writing new blueprints for the cell to change what the cells are doing in useful ways and innovative ways. And synthetic biology, I think by extension, is refactoring or repurposing existing biological tools that you can find in different microorganisms, and then putting them in positions which you can leverage for industrial purposes. I would call that synthetic biology.
What are some common applications that people may not be aware of today of where genetic engineering or synthetic biology are being used in our everyday lives?
Yeah, I think the fastest growing industry, at least, or the fastest growing industry globally, I would say, is agriculture. That's where the most predicted gains are going to be. There's many different places it's used that people don't typically think of, like textiles, for instance. Even these algae I work with, they have these silica walls, and it's also called diatomaceous earth. You can use them as filters, kind of thing, nanotechnologies and things like this.
So you said in agriculture and in textiles, do you have any specific examples in mind of how it's being used in those two industries?
Yeah, so if in agriculture, you can use it to produce medicines in crops, and you can isolate those medicines out.
Like what? Do you have like an example, like
Yeah,
aspirin, or?
so there's this a great historic example would be golden rice, where they engineered rice with vitamin E, and it was a humanity project, and so it allowed people that were deficient in that mineral vitamin.
So rice
to get.
naturally doesn't produce a lot of vitamin E.
Yeah,
And it's
exactly.
consumed largely in a large part of the world where some of the largest populations are. So if you
Yeah.
can get additional vitamin E growing in the rice, you can solve this nutritional issue. So like generally speaking, how does someone do that? I think that's the missing piece that a lot of people who don't come from the microbiology world or biochemistry world don't really like. It seems magical. Okay, how do you get rice to suddenly produce more vitamin E?
It is kind of magical, but so you can identify the proteins responsible in a cell that can naturally produce vitamin E. And once you identify those proteins, you can relate them to a specific gene in the DNA responsible for this pathway. And you can take that pathway out of the cell and put it into a more useful cell, like a bacteria like E. coli, which is easy to produce in the lab and grow up at scale and produce a lot of these things. Or for instance, rice in this case.
That's really interesting. So synthetic biology is really looking at, like, say you target a specific compound that you want to have an organism produce. you can go look at other organisms that are really efficient at producing that compound of interest and kind of look at their DNA and go, oh, they have these instructions that they've evolved over time to be really efficient at producing, say, vitamin E. I can just take those genes out and put them in rice, for example.
100%. And then you can even look at proteins that are optimized for like different temperatures or conditions like pH. And so you can find ones that would be optimal for growing at the temperature rice would typically grow at, right?
Yeah,
So you can optimize for these different conditions, which just makes it so useful.
that's really cool. And that's actually what drew me to genetic engineering, because I was just fascinated by this idea that I could take certain genes out of organisms that have evolved to be really good at doing certain things, and then use that to transfer them into other organisms to kind of complement or bring out those potentials in other organisms. Because every organism is sort of evolved to do different things really, really well. So if you can take the best of both worlds and put them together, then that's a lot of power.
Yes, I agree completely with what you're saying. Yeah, it's kind of incredible. It's like Jurassic Park is finally a reality in a way, but a little ways off.
Yeah. So that's really cool. Have you thought about like where you're, given that you only have a year left, what's
Mhm.
next for you after school? Have you thought about that? Do you want to start a business, or do
I
you?
I feel like my skills lead me most towards a startup kind of environment, but there's a lot on the table, and I'm still kind of figuring that transition out, little
Yeah.
ways out.
What excites you about like what you want to achieve in your career? Do you have some general ideas of like things that no matter what opportunity direction you go, you definitely want to do these things?
Yeah, I definitely want to see algae become a platform for nutraceuticals or as a food source, and kind of as a sustainable food source that could feed the world. That would be my dream come true.
Wow. Okay. You know what I love about people in biology is they're very mission driven and want to do something good for the world. And I like that. You know, for me, that's a breath of fresh air, because I come from fintech, financial technology world, or software engineering world. We're just, not that you can't do amazing things, and there are incredible things that we're using the technology for. But in software development, we spend so much time in our heads looking at bits and bytes and just writing code that we're kind of multiple layers removed from the impact of what our work does on the average person's day-to-day life. And that's one thing I like about biotech, is you're so much closer to life and things that can have a meaningful impact on how we as a species live our lives here on Earth.
Yeah. Also, the community in general is just so receptive to each other and so collaborative that it's kind of amazing. You can meet people so easily, and everybody is just amazed at what's possible and what others are doing because everybody's project is so cool in their own way, right?
Yeah. I get that a lot too, because well, I just did my first transformation of E. coli, which probably for you, you could do that in your sleep, but I just did it from my home lab here, no training or anything. And I hadn't even held a pipette 6 months ago. So
Yeah, I was watching your online Instagram of your transformation experiments. It was very entertaining.
so that was pretty awesome. And I shared it on LinkedIn, and I had all of these scientists who had been working for decades start to message me and be like, hey, I'm a hardcore genetic engineer. If you need help with anything, I love what you're doing. And that's just something that's never really happened to me in software development before. And I think probably because there is that sort of underlying ethos or mission to do something good for the world.
Yeah, I was very excited when I came across your page because I think that's relatively new is these DIY or even like professionals starting to record their activities or their progress, and it's kind of better than a vlog series in a way, you know?
Oh, interesting. You like it more than like sitting down and watching like a full, like a longer format blog.
Yeah, it's like you're doing your
Or
experiments and you hit us with an update, and it's very cool to see that in time progress.
Okay, well, that's good feedback for me. And I think that probably fits in line with where social media is headed. It's very much like spontaneous, like quick hits of dopamine.
Yeah,
So people are
exactly.
like, yeah, we want to just see the quick steps that you do and then the results. And so that
Yes. You take all the hard work out of it for us.
exactly, that's pretty cool. Um, So algae, I mean, well, before we go into algae and the technical stuff, DIY bio, did you, before like running across my stuff and probably other people's Instagram, like have you, and I don't know what it's like in Canada either, but is there a DIY bio movement there or have you heard anything about it? Do you have any perceptions of, I mean, I have my own, like there's
Mhm.
definitely people who are well known in DIY bio that I have some opinions about, but I'm curious to hear what your what your opinions are about the space, what you like about it, some of the concerns you might have if you have any.
Mhm. I think in general, I'm pretty excited at what's possible and the general the general public's perception of what's happening and getting involved and being part of that conversation. I've came across a couple groups in the past. There's a group in the States called BioBlaze I've heard of.
Okay.
And then here in my local community, there's the iGEM teams.
Mhm.
And so we have one of those in our school and they go to different conferences and explain their research and stuff. So I think in general, there's a lot of optimism and a lot of excitement. Here in Canada, also the government is getting very excited about SynBio and there's different groups trying to push that narrative. So it's good to see.
Yeah, that's very cool. I've not heard of BioBlaze. I think you mentioned,
Yeah.
but I'll have to check them out. Um, one thing I've noticed about Canada because one of the sort of sidebar areas of research since I study fungi mostly. That's kind of like my primary organism that I like to focus on.
Yeah.
There is a rise of support and just general public awareness around the potential for psychedelics in treatment. And Canada seems to be much further ahead of the US in this area because there's a lot of actually new psychedelic companies, like people that have engineered yeast to produce psilocybin, which is the primary active ingredient from Psilocybe cubensis.
Yeah.
Have you had any exposure to that world at all? Are you aware of any sort of policy differences between the U.S. and Canada that brings like maybe there's some lessons where I'm getting at is like maybe there's some lessons we can learn here in the U.S. about changing our policy or the way that we work to drive that innovation here in a way that Canada does.
Yeah, I'm not too familiar with Canada's position on like psilocybin or anything. I feel like the public perception is relatively laxed. We recently legalized marijuana, and I know of different companies that are producing THC and like yeast and things like that. So I don't think it's too far off. No.
Yeah, that's pretty cool. What about, I don't know what the differences are, and maybe you don't either, so forgive me if
Yeah.
I'm asking you a bunch of stuff that's outside of your area of expertise. But I've heard that getting a new drug to market in the US is it's a very expensive and cumbersome ordeal. Is it
Yeah.
easier in Canada? Do you guys kind of see the same sort of
I
hurdles and barriers to entry?
I assume it's the same if or very similar. I know in the States it costs a couple $1 billion to get a drug through clinical trials and things like that. But in terms of Canada, we have the regulatory board Health Canada, which we have to go through. And I assume if you're going to go through that process, you might as well go through the state's process because the yields are going to be uh,
Get access to the market
exponential
and
here. Yeah.
yeah, makes a lot of sense. So tell me about algae, because algae is an interesting organism. I know nothing about algae, but I've noticed that in the DIY bio space that it's growing in popularity. There seem to
Mhm.
be a lot of potential uses for algae from alternative foods to I've even seen some really amazing blue dyes produced with algae as well.
Yeah.
So could you give us a high level overview of like what the heck is algae and why is this an interesting organism to study?
Yeah, so algae come in two main big varieties. You have your seaweeds and then your microorganisms, which are just single-celled algal cells. I work with the single celled, so I have more knowledge on those. They're a very diverse class of organisms. Many, there's estimates of hundreds of thousands of species. And
Wow.
the algae I work with, up to 35 to 50% of its genes, the ones that encode the proteins for different functions, have unknown function related to them. So it's
Wow.
this huge Pandora's box of what's capable and what don't we know yet and what can we utilize. And beyond that, the perspective for DIY bio is they're very or relatively easy to work with. They're a bit slower than different model organisms like bacteria or yeast. For instance, with yeast, you're working on the time scale of a doubling every hour and a half of the cells from 1 to 2. With the algae, you're looking at every 24 hours. So you have to kind of plan your experiments a bit more and what you're doing. Be more a little cognizant, I'd say.
Yeah, that it kind of reminds me of fungi in that way. I started working with culturing fungi, and unless you're working with certain molds, Ascomycota, those are typically pretty fast growing. But by and large, a lot of fungi are kind of slow to colonize and grow on just plain media, regular like PDA or MEA media. But then when I started doing E. coli, like things started to move much quicker, much quicker. And so for me, starting with fungi and then doing E. coli, actually E. coli seems a lot easier because things move so much quicker.
I'd agree. Yeah. We worked in 2017, 2018 with a fungus called Meshnickovia. And we were trying to develop a transformation protocol for it because one of the researchers at our university, it was a collaborative effort. He discovered this specific strain. It turned out it used an alternative genetic code. So the language it was using compared to other cells was different by a couple letters. And so we had to recode all of our selection markers for it to be successful.
Wow. So I would love to dig into that because all of the work that I'm doing right now with transforming E. coli, I'm getting ready to do my first mini prep kit. For people who are experienced, they're like, oh, they're probably rolling their eyes because they do this stuff every day in their sleep. But I'm working my way up to, I have captured probably 50 different species of mold and fungi from my local environment. There's not a lot of published transformations of filamentous fungi. And so that means there's a lot of opportunity for research and to sort of be on the cutting edge of figuring out how do I get exogenous DNA into these organisms, because no one's really like worked on this. And there seems to be like several different approaches, from the polyethylene glycol approach. There's electroporation, kind of shock the exogenous DNA into the cell. And then there's the agrobacteria mediated approach.
Right.
Given that you worked on that project, I kind of want to pick your brain a little bit because I'm leaning towards using the agrobacteria approach to to start to make headway into this area. Do you have any thoughts on which like pros and cons with those approaches and which one would be good to start with?
So my supervisor would probably murder me if I didn't say conjugation using agrobacterium would not be your way to go. That's kind of our lab specialty and what was his claim to fame, I guess. So when he worked at the Craig Venter Institute, he developed a protocol where bacterial conjugation to the algae cell for the first time as a transformation method. And so if I didn't say trying to use conjugation to first transform these yeast cells, or not yeast cells, sorry, fungal cells, then I'd probably be a little off base.
Is that, so that's the first time I've heard the word conjugation in this context.
Okay.
Is that using the agrobacteria to transfect the fungal cells?
Yeah, so the agrobacterium, it harbors a plasmid, which contains all the proteins required to move the plasmid, which is just a piece of DNA that's outside of the chromosome, from the donor cell, which is the agrobacterium, to your fungal recipient cell. And so it contains the genes to create this pilus, which reaches out and touches the recipient cell and brings them into contact, creating like a cell aggregate and allowing for this DNA transfer to occur.
That's pretty cool.
And that whole process is called bacterial conjugation.
Okay, cool. So that's good for me to have those keywords, because that lets me, when I'm doing searches for papers, to know what to type in.
And then to add to that complexity, there's different conjugative plasmids, which are those extrachromal DNA fragments. They're circularized inside the cell, and they have different machineries for causing this process to happen, which leads to different groupings of them called incompatibility families. Because if you try to put two of the plasmids that are incompatible together, then only one can kind of be replicated and moved.
Oh, interesting. So you really touched on something that I haven't figured out yet, but it's something that I need to figure out, which is like, let's say I take one of these, and I have cultures all around me here. This this is a Aspergillus flavus. And
nice.
this is actually probably, I would consider, one of the model organisms, because Aspergillus, there's a lot of papers on doing transformations in Aspergillus. So I want to kind of walk through sort of a technical problem, and maybe you could at least kind of help me and then other people who are listening kind of figure out, how do I attack this problem? When we're talking about doing a transformation, we're talking about taking these circular pieces of DNA and transferring them from bacteria into the fungal cell walls, and kind of just using the natural bacteria's natural mechanisms for cloning itself and making multiple copies of that plasmid DNA. And then, and as you said, the plasmid DNA is encoded with special proteins that allow it to reach out and transfer itself over to the fungal cells.
Yeah.
How do I go about, let's say I want to do a transformation of a fungi that's not been published. How do I find a plasmid DNA that will work for trying to do that transformation? Like, I think it's called a backbone. How do I find a backbone that has a good probability of working? And in the beginning, all I want to do is maybe just do GFP to one of these new species that hasn't had any documented transformations.
Right. So that whole process is called domesticating the organism, making it easily genetically engineerable. And there's a couple problems that you'll have to overcome. First is identifying some key components of that backbone that will allow it to be stably maintained, replicated, and propagated in your host organism, where you're trying to get that DNA to go. So the first is like a centromere. So allowing it to be segregated between cells, origin of replication. So a specific DNA sequence that allows that DNA fragment that you're putting into the donor or recipient cell to be replicated, as well as a selectable marker. So that is like an antibiotic resistance, or GFP, like you said, which is a positive selective marker where you can observe it instead of selecting against ones that don't contain that DNA. And in order to express that GFP, you need a promoter and terminator that would be recognized by it. And so I think the easiest way to get at those elements is finding the most closely related species that has been transformed or with a backbone, and start there and then work backwards and try to mine the genetic information for likely sequences you could use.
That's what I was planning to do. There's other fruit body bearing transformations that have been done on things like the button mushroom, for example. So if I have another fruit body bearing mushroom and that hasn't had anything documented in terms of the transformation, it would make sense to use a construct very similar to what's already been done to that similar species, even if it's a totally different species. And I've even noticed that, like, even for cross species transformation, there's still a lot of overlap. Like, many of the transformations are using some of the same promoters that are in Aspergillus, which are just like a totally different species as well. So there does seem to be some transferability in the species in terms of the underlying components that you can use to make the machinery work. One question.
Go ahead.
No, go for it.
If I could just give you a quick example of when my PI was making these transformation backbones for P. tricornidum, the algae. He he cloned one of the whole chromosomes in pieces with his backbone that he used to clone it in S. cerevisiae yeast. And so with walking down the chromosome, he was looking for the genetic elements that would allow for the replication and segregation of that plasmid being transferred to the recipient, which is P. tricornium. And by doing that, he actually found that the centromere he was using to clone in yeast, his negative control, was one of the few that were actually working, resulting in transformance.
Wow. Is that process published in a paper or any place?
Yes, I can link that to you. It is the creating designer conjugation with designer.
That'd be
It's a
kind of cool.
2015 paper.
And I know it's on algae, but this idea of kind of walking through the chromosomes, there's a strategy here where at the heart of every organism, there's a bunch of DNA inside of chromosomes. There are some systematic ways that you can invoke different sections of the genetic material and test what it is that they do. And I think when I'm working on my own research here of like taking one of these species that hasn't been, did you call it domesticated? I think is what you said. They haven't been, I'm learning the terminology as we go. They haven't been domesticated. I think that's probably part of the work that would end up going in my own published research, which is this exploratory work to kind of figure out, what unique characteristics about this organism exist that play a role in successful transformations of the organism.
Yeah.
And so those techniques are published. People have done things like that. It's helpful for me to go and read and piggyback on some of the work that they've done.
Do you find doing DIY bio, you have any trouble accessing those articles or anything like that?
Yeah, it's a good question. So PubMed from the NCBI database has a lot of stuff, but frankly, I'm doing what a lot of DIY bio people do, and that's using Sci-Hub to find papers and read them. And in the beginning, when I first started looking and reading these papers, it was all like a foreign language to me.
Yeah.
But just like everything in life, when you expose yourself to something that's completely foreign over and over and over again, these patterns begin to emerge. And I've even noticed that if I'll go pretty heavy in doing research and reading papers, really trying to read it critically and understand what it is, and a lot of looking up terms and bringing that back to my reading to try to make sense of what it is that I'm reading. And then I'll take a break. Because I think what happens is that new information integrates in the mind. It kind of just sits there. And when I come back and read the paper, it makes a lot more sense after having spent some time. And so people don't know, like, research papers they have a very similar layout. There's usually an introduction. There's usually like a summary of the findings. Then there's a result or a tools and methodologies section, which actually walks you through the steps, exactly what was done, setting up the experiment, what strains were used, what reagents were used, what protocols were used. And I think the challenge for DIYers is there's a lot of pointers to other papers. And so that can be difficult to keep track of, because you'll be reading a paper on a transformation and it'll say we used Wang 2016's protocol. You're like, okay, then you got to go click on Wang, and then you read Wang's, and it's a totally different context. And then that paper has like 50 different references out to some other place. Right. So like part of the work is like coalescing all of these information sources into like a singular place. Yeah. And also I think a challenge for DIYers, you don't know in the beginning how important some components are and what areas can you make trade-offs with. I'm in a less than ideal setting here. I'm not, I'm not in a clean room by any means. So I have to take very special care to
not contaminate all your samples and things like this. Yeah,
exactly. And then I'm trying to do things DIY, which means I'm not using always lab grade reagents. And so it can be done, but that's part of the fun, but also part of the challenge of doing things DIY style is you can waste a lot of time pretty easily because you don't have like that professional guidance or mentor. That's one of the trade-offs. On the other hand, you have this awesome opportunity to do science that is not being done by a lot of people in this sort of fashion. And the DIYers, the amateur community, because they're under those constraints, they often will encounter really clever ways to do things that might not have ever been considered in a more formal lab setting. I think all of those things working together really help progress science forward and make it more accessible to people.
I think another big thing with the DIY is you have kind of total freedom on whatever you want to research or whatever species. It doesn't matter if somebody like a funding agency doesn't think it's relevant or is ever going to give money back to society, and you can really look at whatever you want and then show them that it's relevant.
100%. And that's a blessing and a curse because the world is so wide and it's like hard to stay focused on one particular lane. But that's part of the learning journey. There's a whole strategy around how to manage that. One thing I wanted to go back into is you talked about the plasmid needing a origin of replication, and that's something people will often see when they're looking up like plasmid maps, or they start to think about constructing their own DNA plasmid that they want to put into another organism. It has this section called the origin of replication, which is kind of what tells the organism, it taps into the organism's own mechanism for consuming that DNA and replicating it. Is that right? Is that a good way to say it?
Yeah, it's a sequence. It's a genetic sequence recognized by proteins in the organism, which allow it to be maintained and replicated faithfully across generations.
So my question is, if I'm moving a plasmid DNA from agrobacteria into fungi cells, do I need two origins of replication there?
So it depends on your experimental design. There's different ways to go about it. I think with agrobacterium, you have what are called border sequences beside the construct that you want to put into your recipient cell. And so the genetic machinery on the plasmid will cut out that sequence to be transferred. And then that sequence can actually integrate into the existing chromosomes. For instance, your fungi of interest. And so that could be stably maintained by hijacking off of the machinery or the genetic elements already found in the chromosome.
Okay. So it's kind of a way of like hacking into it a little bit.
There's like advantages and disadvantages for chromosomal integration, and there's advantages and disadvantages of maintaining an episome or a plasmid in the recipient cell.
Got it. So going back to algae, were you attracted to algae before you started to do the research and to dedicate your education to it, or did you find
I found out about these things and it took me forever to understand exactly what was going on. I thought they were plant cells at first, but they're very different from plant cells. They undergo endobiotic or symbiotic relationships with different bacteria, so they require bacteria to exist.
Wow.
Their chloroplast, for instance, has evolved multiple times. It's by consuming other algae cells and maintaining their chloroplast inside of another membrane. There's just so many different things about them that make them interesting and amazing, and that's what made me attracted to them after I learned what they were.
When I think about algae, I think about when I was a kid, I'd have a fish tank and I always had to do the cleaning and to scrape off the algae around the edge.
That's a good point. Yeah.
What I didn't know, and I just learned something now, is there's actually bacteria in that water that's necessary for that algae to grow.
They're actually relying on it for the vitamin B12, and that's the most expensive component that we put in our medias to grow them up.
Interesting. So I don't know where that bacteria comes from, maybe the fish or something. It kind of like, or maybe just lives in the water or whatever, but it produces B12. It sort of metabolizes it and creates it. And then the algae is like, oh, hey, I need that B12. How does algae seem to just pop up into existence from, let's say, like a fish tank? Are there algae cells everywhere like there are with fungi cells?
They're everywhere. There's heterotrophic strains, so they just exist off of carbon sources, and they can live in swampy areas. They can live in the dirt. They can live in the water.
Are they in the air?
I don't believe many are in the air. Because they're eukaryotic cells. They're probably a little too heavy to be floating around very often.
Got it. So I'm just thinking like the fish tank example, because I think like for people who don't know about algae, that is probably pretty familiar to a lot of people is if my fish tank keeps getting algae growth on it, where might the algae cells have come from in that case?
So I would predict that they were just in the water source or from the marine life that you put into the tank.
Got it.
Or if you get some stones from the swamp or anything like that, or the lake, then they pop up. Some of the algae are even actually bacterial cells called cyanobacteria.
Oh, I see.
So you can even have algae that are bacteria.
Are they more closely related to bacteria than they are any other organism?
So there's a grouping of algae called green algae, and that's the lineage that led to the land plants. So they're pretty related with land plants, but different algae are closely related with different things. It's such a diverse group of species that you can almost point in any direction.
That's really, really cool. And you said there's thousands of species of algae, and I wasn't aware of that. That's pretty cool. What work is being done today to tap into the potential uses for algae?
There's tons of groups internationally working on different algae. There's one group I know of that's taking metagenomic DNA or sequencing data. So they take a sample out of, for instance, the ocean. They extract DNA, they sequence it all, and then they just start identifying species that haven't even been identified because they can't be cultured. So it's a way of mining all this information without even ever seeing the species that you're getting it from.
And how come they can't be cultured? Is it just because there's things that are happening in nature that are very difficult to replicate in a controlled setting?
There's just different variables that we probably don't know, or essential nutrients or minerals that we're not adding into the typical medias we would use to culture things.
Got it. Because there is like fungi that's the same thing, that there's lots of them. Many of them are very like different kinds of truffles, incredibly difficult to cultivate. That kind of drives the price up really high and kind of plays on the, some of them even require that ants, like there's these special ants that build an ant nest, and they play a role in helping this fungi grow. And it's been impossible to cultivate this one species because we don't yet know how, but the ants somehow play a role in creating the right environment for them. So that's pretty cool.
Nature's pretty incredible. There's so many things out there that are yet to be identified or understood, and the chance that they'll be useful in biotechnology is so high. It's a great opportunity for anybody getting in the field.
And I think what you said with the metagenomic sequencing, that sounds very similar to the DNA barcoding stuff that I've been like embarking on with fungi and sharing with my network, where with fungi, what we're doing is we're looking at the small subunit section of the ribosomal DNA, and that has a highly variable section in it is a well-conserved section in it that doesn't changes subtly enough between species that you can make these linkages between, and you can identify them. You identify the species, and you can generally taxonomically figure out the distance between its other more distant species or even really closely related ones.
Yeah, the ability to sort them out and kind of group them in a meaningful way. It's super helpful for sure. I know they also do that with like the mitochondrial ribosomes, because they change so slowly as well.
Yeah, that's pretty cool. And what you were saying, like people going out and finding algae in the ocean, is that what's known as bioprospecting, where people are kind of going out and trying to find these new species or new characteristics?
So that's a pretty unfamiliar term for me.
Yeah, I've seen a couple of people use it. And I think what it is people just going out into nature, trying to find new species or trying to find strains that have some unique characteristic about them that could end up being useful.
See, I think that's the dream. Get some government funding to go out into nature and find some very unique, interesting organisms and then just study them.
Go into like a volcano, walking around the volcano and be like, oh, there's some algae growing right here by this vent.
There we go.
That sounds like a ton of fun. So you're exploring algae, and you kind of mentioned one of your goals was like perhaps use algae as an alternate food source.
Mhm.
What makes algae kind of a prospect for that?
So there's already nutraceutically, if that's a word, uh, they have many properties that are already healthy. So they have different pigments which help with cardiovascular health. They have healthy fats. There's different possibilities to express things in these organisms, and that's what makes them so beneficial as a food source, I think, as a potential food source.
We're talking
I
about one species in particular?
So the species I work with, Phylodactylum tricornidum, is approved, I think, by Health Canada and FDA for consumption. But in general, a lot of these algae, because they have such diverse metabolisms, each one's kind of have unique traits that make them more useful than others.
Interesting. And I didn't know that there were pigments that can impact the cardiovascular system.
Yeah, I forget exactly what they're called.
When
They
I
have the
think
suffix.
pigment, I think of a cell that generates a color.
Yeah, so this is the pigment that gives the cells a brown black color.
Is that melanin, or is that some other sort of pigment? Don't worry about it if you don't
Yeah.
know it, but so it doesn't matter, because at the end of the day, a pigment is a chemical. It's a compound.
Right.
And
It,
so,
it, it has some photospectral or optical changes when it interacts with light.
yeah.
So
Yeah. And then apparently somehow that plays into our cardiovascular system.
yeah, I didn't read too much into it. I saw that was one of the highlights with this algae, P. tricornum.
That's pretty cool. And you said that they have like a diverse metabolism. Does that mean that they can feed on a wide variety of different substrates?
So what I mean by that is they have different biochemical processes inside, and those which would be not usually seen in things we're used to, like animal cells. So like the ability to metabolize silica. So they have biosilica integrated into their wall and gives us this glass structure, for instance, in
Wow.
the algae diatoms. And that's the ones I work with.
I mean, that sounds like, could we create a algae recycling plant and throw a bunch of glass in there and have the algae eat it?
Right. And then
Theoretically?
you could repurpose them for nanotechnologies, because these biosilica walls are used to create different symmetrical structures. If
Oh,
you look
wow.
up some of these diatoms on Google Images, they have very regular and symmetrical shapes and things that could be used for moving liquids around and things like this.
So you're saying potentially we could grow a lot of algae. They'll grow into this crystalline structure that we could, I mean, we might end up having algae-based bottles, for example.
That could be very cool. I should get my idea book and write this down. No,
That
that
sounds
sounds
awesome.
really interesting. Yeah.
I had no idea. So like there's algae that will just take raw silica and then digest it and then produce.
So it has to be in the correct form for it to be able to uptake it and process it. But yes, they form these structures in these compartments inside the cell, and they just push them towards the edge. And when they divide, they have these two half valves that separate and just get smaller and smaller until they make a larger cell again, go to a different state.
That's really cool. That's something, what thought that comes to my mind is when you're just sort of on the ancillary edges of DIY bio and hearing about some of the things that can be done. For example, there's plastic eating fungi and plastic eating bacteria. But then when you dig into it, you realize, okay, this is theoretically possible, but there are some constraints that make it difficult. It's got to be the right kind of plastic, and it's got to be like, there are these parameters that end up making the feasibility of it still pretty challenging today.
Mm-hmm.
And do you think like there's a lot of the work and research being done right now is trying to figure out what those parameters are to fine tune them so that you could really open up these applications?
Certainly, yeah. I think there's a big difference between the practicality of things and the theoretical. I think finding something out and thinking it could work versus actually scaling something up and the problems that are associated with that are very challenging. And I think there's a lot of people in the community that are trying to standardize parts, things that can be moved between systems and that have predictable functions to help with these things, but it's difficult to for instance, engineer a pathway that contains many different components, and you're trying to control how much of each component is present at a specific time, or different toxicities that happen when you're trying to really express a protein to high levels inside the cell. There's lots of problems and challenges that come with making something useful versus discovering if it's possible.
Yeah, that makes complete sense to me. One of the things that I hear VCs and life science investors talk about is helping make that transition between a researcher to a business person or startup, making that easier. And I kind of just wanted to get your feedback on that. Do you find, I just find with anything in life, you can go really, really, really deep into one specific area, but then you kind of lose this other world that's kind of around you because you're so laser focused in deep into this one particular area. Then again, if you pull out and you're too broad, you're going to lose the clarity of having that focus into one area.
Yeah.
Do you
It's a
think
difficult balance?
Yeah, I was going to say, is that something you think that other researchers like yourself are like struggle with, which is how do I take this idea and transition that into a business? Maybe you could talk a little bit about some like why those challenges exist and things that could be done potentially to help researchers make that transition.
So, at least from my perspective as a graduate student, the program that I had entered had a very large emphasis on making sure things are practicable, as well as they give different courses like translational research that help you kind of gear your research towards going as a startup avenue, and what that kind of looks like and the steps you have to take, and patent law, things like that. Specifically, my supervisors are, one of them has filed a couple different patents, and the other had a startup which he had here based in London for a while called Designer Microbes Incorporated. And so
Good name.
I have, yeah, right. I have some supervisors that are very much focused on making things practical and useful.
That's great.
And so they emphasize that throughout my degree so far.
Yeah, so they're like when you're proposing doing your research or something like that, they're helping guide you towards something that might have some commercial viability at some point.
Yes, and specifically because I'm in the School of Medicine and Dentistry, Schulich, here at Western University, it's very much how are you going to relate this medically? How's this going to be relevant to anybody medically?
Yeah. Can you talk a bit about the research you're currently engaged with right now?
Yeah, for sure. So the overarching theme of my whole project was to develop this biotechnology to allow easy engineering and delivery of whole mitochondrial genomes in these two algae, Phyllodactylum tricornum and Thalasseria pseudonana. And so the way I broke up my project was, first I had to clone them and show that I could manipulate them and use the model systems I was working with. And then the second is developing selectable markers and things to transform the mitochondria, those protocols. And then third is delivering a whole mitochondrial genome.
Okay, so I want to break
something useful. Yeah,
I want to break this down, because I think it will be helpful. I'm working on at a high level, we're talking about moving DNA around, right? You're going to design some DNA and try to get it into these two species of algae.
Yeah.
When I think about this, I think about a plasmid construct, which has those key components that you mentioned before: origin replication, some sort of selection marker, maybe a visual marker as well, like GFP, and then maybe your multiple cloning site where you have your gene or genes of interest kind of inserted into that. All right. How is that different than whole genome mitochondrial DNA? And what's the application of getting that? Like, what's the benefit of getting that into the algae?
So the idea is, if you could develop this technology to deliver a whole mitochondrial genome, in a perfect world, very far away from now, we could build a cell from scratch and import this whole mitochondrial genome in and kickstart this eukaryotic cell up to be doing useful things.
What's a mitochondrial genome? Like, how's that different than regular DNA that might be found in the nucleus of a cell?
Right. So the nuclear DNA is typically very large megabases on scale. The mitochondrial genomes are around 50 kilobases, 50,000 base pairs to 150, at least in the species that I'm working. Uh, there can be a much larger range if you look at things like plants and things like that. But
And this is
specifically,
DNA that lives in the mitochondria of the cell?
yeah, so it exists inside the mitochondria. It's often associated with the inner membrane, and it has a higher ploidy than the nucleus.
What
So
is
it
ploidy?
exists. Ploidy is the number of copies.
Oh,
So
okay. So
so
what?
you can get more expression kind of thing. You can have 36 times the expression
So
if you were to.
I don't think a lot of people realize that DNA is found in different areas of the cell. I mean, certainly most people know there's two different kinds of cell with and without a nucleus,
Mhm.
but there's also DNA in the mitochondria of the organism, which is sort of the powerhouse of that is used for metabolism, as you said. And that's obviously important in biotechnology because a lot of the applications are trying to get to tap into the cell's ability to produce these metabolites.
Mhm.
So if you can control that, you obviously can do some pretty powerful things. Go ahead.
Yeah, the people know the mitochondria is the powerhouse of the cell, but it does have additional functions like fatty acid metabolism, beta oxidation, which creates energy, as well as amino acid synthesis, which is creating essential amino acids for the cell. There's multiple functions and metabolisms which you can manipulate the mitochondria to affect the cell.
Why does the DNA live in these two different places? You know, like
Yeah.
it doesn't move from the nucleus because obviously when I think of nuclear DNA, I think like that's the DNA that even encodes for the mitochondria itself. So
Yeah, so
when does DNA end up in the mitochondria?
the hypothesis of how the mitochondria came about was there was an archaeal ancestor cell which engulfed predicted an alpha proteobacterium. It doesn't really matter. Engulfs a small bacteria which somehow escapes degradation inside this cell and becomes its own organelle. And then over a long period of time, these large pieces of its genome move to the nucleus, and anything that's duplicated it just gets rid of and keeps all the useful stuff. And eventually, you get the system to import the proteins from the nucleus into the mitochondria, and then you start developing these signals, these transit signals or localization signals to traffic proteins from the nucleus into the mitochondria. And so proteins which could not traffic remained in the mitochondria, as well as proteins which were important for regulating redox potential or acting as a scaffold for larger protein assembly. Those types of genes remain inside the mitochondria.
Wow. Okay. So when the cell is being formed and growing, does DNA does it migrate from the nucleus into the mitochondria as the cell itself is being developed?
No, so the nuclear DNA and mitochondria divide separately. They communicate to each other in different ways, but the mitochondria divides by
Wow.
itself.
Wow, wow, I had no idea. So is there some other process in place that's decoding the mitochondrial DNA? Is the ribosome playing a role in that as well?
So the mitochondrial DNA has all the machinery required for transcription and translation, the
Wow.
expression of those genes inside, and
So my mind's blown. I had no idea.
they use a different system as well. Like they use a bacterial ribosome compared to the eukaryotic one in the nucleus.
Wow. Okay. So it's almost like its own like proto-nuclear, like
It's like
proto-nucleus in a way. It's like it's an organism within an organism.
I like to imagine as a very like strong, intense symbiotic relationship of like this pseudo organism living inside of another.
And so the mitochondrial DNA has in like I think of things in terms of code. So I might say things like instruction set, right?
Yeah.
It has the ones and zeros, but it's really A, T, G's, and C's. So that it has specific instructions that don't also exist in the nuclear DNA.
For the most part,
For the
there's some cases where there's some existing in the nucleus and the mitochondria.
Gotcha.
It's,
But it has a lot of instructions on how the mitochondria should perform and function.
it maintains maybe 15 genes, at least in
Oh,
my system.
okay.
But importantly, they have what I would say is like different operating systems.
Oh, I see. Okay.
So the means by which they express the mitochondrial genes and translate them would be similar but slightly different than the nucleus.
So is it still double-stranded?
Yes, and
Still
it's sometimes linear, a lot of the time circular. There's different structures. Kinetoplasts have these like interlacing rings, and they have mini and maxi circles, what they're called, and
okay.
it creates this chain mail kind of looking thing.
Wow, cool. And so going back to the original question, now that we have like some context and understanding of what the heck's going on, why would we want to do whole genome insertion or transformation of a cell of its mitochondrial DNA? We want to have, you said there's 15 genes. Does this give us a lot more control over the cell function?
Right now, it's very difficult to manipulate the mitochondrial genomes. If you could do a whole mitochondrial genome delivery or even a replacement, removing the one pre-existing there and replacing it with yours, you could, for instance, recode the genome to use different codons, the things that are read. And so it's a kind of a form of biosafety. Things that escape wouldn't really make much sense to the natural life forms. And second, you could easily insert whole biosynthetic pathways. So I have my mitochondrial genome in Cerevisiae yeast and E. coli, and I can easily manipulate it and add, say, 15 genes, and then move that whole molecule into the mitochondria, install it. And then the mitochondria, when you express things in organelles, you tend to get higher yields for different reasons. And so there's a huge benefit to using the organelle.
That's so fascinating. I had no idea. So right now, a lot of the work is figuring out how to do that, the transformation in and out of mitochondrial DNA, because it sounds like if you can kind of master that technique, the world opens up to you in terms of being able to harvest the cell's ability, its function, cell function.
Yeah.
And so it sounds like there's things that you can do right now by modifying the nuclear DNA, but maybe it's less efficient and more complicated than if you were just able to go directly into the organelle itself.
Right. So like the mitochondria, it has some advantages like compartmentalization. So if you're expressing your pathway in there instead of the nucleus, you have less competing reactions with the cell and you're fighting for less substrates and stuff.
Yeah.
Additionally, you have higher concentrations of your proteins that are turning over your substrates into product because you're working with such a small compartment inside the mitochondria. And as I
Oh.
said, that ploidy, because you have multiple copies of it, you have multiple copies of your construct inside of there and multiple units of it being expressed. So you can get higher yields. And at the same time, reducing that toxicity because the rest of the cell doesn't see these proteins.
Wow, that's pretty cool. I probably at some point in the next generation will probably be some transformation of both so that you could just really maximize both systems in tandem with one another.
I hope in the future we can make a whole eukaryotic cell from nothing and
Well, yeah.
install these genomes.
And you mentioned that's where you're going. And I have read about a couple of efforts to build a cell, which is like, let's deconstruct the components of a cell and figure out how to create an artificial cell. It sounds like that's where we're headed.
Yeah, I went to a couple conferences in America called the Build a Cell. And
That's where I heard of it.
Yeah, they work on what are the limitations and challenges and which of those can we tackle short-term, mid-term, and long-term kind of thing. And then how can we get money from governments to push these efforts forward?
Yeah, that will be a crazy world to live in when we have these synthetic cells, because it's kind of hard to wrap my brain around what that means. Are they machines? Are they real? Because they are functioning, living chemistry, working in coordination.
Is it right? Is it wrong? The morality of it? It's all kinds of.
That's interesting too, because who knows how those cells will be used. But I think all of this is inevitable. I think to some degree it's moving slow enough that we have time to adjust and think about these problems. And if it's going too fast, maybe there's an argument there to slow down a little bit so we can kind of hash some of this stuff out.
Yeah, I agree.
On the topic of just creating a cell and life and DNA, we just had the Perseverance rover land on Mars, and one of its big missions is to search for microbial life on the surface of Mars. And I think, when I talk to people in my circle, which most of them are not connected to biology or life science at all, they don't understand the significance of finding life on another planet, because when I explain to them, like, DNA is inherently unique to Earth, like how life comes together and the central dogma, but it's very possible that in Mars or other parts of the universe, life came together using a totally different tactic or methodology. And if we're able to capture that, that may reveal, such as building a cell, we may discover new ways of how life can organize itself. And I think that's fascinating. Do you follow that at all? Are you excited about that?
Slightly. I was in the lab the other day, and my supervisor came running in with the live stream saying, "It's about to happen. It's about to happen." But we work with this microorganism called DRAD, or D-radiodurans. And it's super cool because you can hit it with all this UV, and it just decimates its chromosomes into small little fragments. And then it just takes them all and puts them back together. And you can watch this happen over time. And it's just incredible. So
it's like some kind of repair mechanism.
Yeah, it has such a high number or copies of it. It just has all these templates to repair itself, and then it can fix its genomes. And so one of the graduate students in our lab, Stephanie Brumwell, she works with that, and she's trying to make a system, I think, to help, what's it called, terrestrialize, make the soil on Mars more livable.
Oh, that's interesting.
Right. So there's also that approach. In addition to finding those life forms, converting them into more of our environment,
infecting the planet with our microbes.
Yeah, I think finding life forms that are so different than ours will really help open our minds on what is possible and what we could make here on Earth as living systems.
Yeah. I mean, I just get excited about the idea of like a sample coming back to Earth, and then we try to, like, I don't even know how, I guess we try to first look at it under a microscope is probably the first thing we would do to, like, does this thing even have a nucleus, and does it even have chromosomes?
Yeah. I think there's still even the discussion on what we could classify as life, or what people could agree on. Is this even a life form?
Like, that's a great question too. Like,
they can't even agree on what a virus, if that's alive or not, right? So
yeah, that's a good point. At some point, when you get down to the elemental elements of a thing, it's sort of like, how does consciousness emerge from just matter, or is consciousness its own thing somehow?
Yeah, does it exist outside of this? Yeah.
Yeah, and that's a whole other philosophical, like, conversation, but it is very much related to this idea of, like, what is life? And when you get down to, it's like, it's all the same elements in the periodic table. You just arrange them in the right way, and they have these functions.
Yeah, like, does it just matter that there's some sort of turnover or reaction happening, and that eventually that reaction leads to enough accumulation that divides into two similar things? Is that life? I don't know.
It's a fun thing to sort of do these thought experiments with. It's really just like kind of amazing. So how is your research going, and have you made, like, some headway into this idea of whole mitochondrial genome transformation?
Yeah, so I finished kind of the first phase of my thesis, which was cloning these genomes in my model systems, E. coli and Cerevisiae, bacteria and yeast, and showing that I can engineer them pretty quick without DNA synthesis and create new iterations of it.
So you were able to take mitochondrial genome DNA and manipulate it in some way, and then have a bunch of other yeast clone it, basically make copies of that same.
Yeah, so I extracted the DNA from the algae cell, and then in one approach, for instance, is called TAR cloning, transformation associated recombination cloning. And you take the whole mitochondrial molecule, and with a unique cut site, you break it open, and you develop that vector backbone, which you had talked about, with homology, to where
Oh,
directly
it
adjacent.
attaches onto the end that you use to cut.
Right, and then you transform that into yeast, and yeast through homologous recombination and repair will put those two fragments together, and you have a whole mitochondrial genome with your backbone inserted inside of it.
Wow. Okay. So I guess you'd say they ligate those ends together.
Well, not exactly ligation, but through homologous recombination.
Okay.
And then after that, I used PCR to amplify the fragments with homology to each other. And with 10 of those fragments, by PCRing them, you get a whole bunch of them. And then again, you can transform those into the yeast and clone another version of the mitochondrial genome. So if you wanted to insert something with one of the fragments, you would just leave out the homology and add your DNA to be inserted with the homology to where you want to insert it.
Got it. Just add your DNA with ends on them to match with where the cuts were on the underlying construct. Makes sense. How do you take, just like high level, when you extract the mitochondrial DNA from the algae cell, how do you separate that DNA from the nuclear DNA that comes from the cell?
Yeah, so the main way that this was done in our experimental design was through that homologous recombination. We isolated all the DNA of the algae cells in agarose plugs. It's another method, but it keeps everything stabilized, and so that way nothing breaks or shears and snaps.
So you grow a bunch of algae, you create these, grow them on agar media.
Well, you grow them either on agar media or agar media, or in liquid, and then you take this low melting point agar, so it's not so hot, it's like 50 degrees, and it captures this highly concentrated number of algal cells just inside a very small agarose plug.
Got it. So you kind of create this dense collection of algal cells in a plug,
and then you just blast them with aggressive buffers to blow the cell up, essentially, and wash everything away.
Okay, so that's not too unlike how, like, I'm doing the mini prep, which is using enzymes and lysing the cell to release the DNA. So you're kind of doing the same thing.
Yeah, exactly.
And so, but now you have all of this DNA mix and match between the mitochondrial and the nuclear DNA.
And so when you transform that into the yeast, you're transforming some of the nuclear, some of the chloroplast DNA, some of the mitochondrial DNA into all these yeast cells, but only ones with whole mitochondrial DNA, as well as your vector backbone homology, will recover a circularized plasmid because of that homology sequence.
Because the way that you got it, and there's a missing link here that I'm missing, and that is you must be doing something to prepare the yeast cells, like you're enzymatically cutting them or something, so that you can then paste in this.
We're giving them what's called a spheroplast treatment. So you hit them with these enzymes called zymolyase and HLZ, lysozyme. And so it degrades the cell wall, and you're trying to catch them where, if you put them in sorbitol, which stabilizes them, they're not blowing up so much, they're not popping. But if they go in water, the osmotic change the difference between the solution and the cell causes the cells just to lyse. And so you're looking for this happy medium where they're not blowing up too much, but they've lost their cell wall. And then that allows for the DNA to more easily get integrated inside the cell.
Cool. And so that's part of the research that you've been doing, which is you've got to find these titers for how much of the sugar and how much, so that you're maintaining this delicate balance of, you don't want to completely like destroy the cell altogether.
Yeah, a lot of these methods were kind of optimized previously, but putting it into your own hands and trying to get that experience and learn how to do it, it's difficult. I think the best in our lab so far is 2 tries at spheroplasting before a successful transformation.
Wow. Okay. Well, that's good. That's a great data point for me, because I could probably do that. Probably multiple orders of magnitude is how much longer it'll take me to
DM me and I'll help you out.
Yeah, for sure, man, for sure. So, okay, you've got another year of research. Sounds like you've already got 2 manuscripts. It looks like you're well on your way to doing some incredible work in your career. And so I'm really excited to follow you on that. What are, just like right now in your world, what gets you excited? What are some of the research projects either going on around you or just in the world that make you optimistic and excited about where the world of synthetic biology is headed?
We have a lot of great team members in our lab with just incredible projects, and they're doing incredible things. I don't know how much I can say about their research without kind of giving too much away, I guess, but there'll be very interesting things coming out of the Karish lab soon. I know I should have another paper later this year, maybe in the fall, and it'll be related to conjugation and really making a better process to conjugate to different types of cells and things like that. So in terms of my thesis, that helps progress that DNA delivery method, as I was saying. But worldwide, what I'm most excited about, I think, is all the different startups and creative people taking on different challenges and using synthetic biology to solve them, as well as the international collaborations like Build-A-Cell, which are doing really incredible things.
Well, that's very cool. Ryan, thanks so much for being on the podcast. I really got to geek out and nerd out on some of the more technical stuff, which is a lot of fun. So I really appreciate you, diving into that stuff and helping educate a layman for a lot of these things. Super, super awesome to talk to you and to hear about the research you're doing and to learn about algae. You kind of blew my mind with the mitochondrial DNA. I didn't know about all of that. So that's pretty awesome. And I think I think a lot of people don't know about that. So hopefully they can learn something from this and get excited about it and start to follow that emerging field.
Hopefully we get the chance to talk again. And I look
I would love to
really promoting it. Yeah.
Yeah, absolutely, man. All right. Well, have a good one. And I appreciate you being on the show, man. We'll definitely talk soon.
Thanks, Josh. Bye.
All right. Bye-bye.